So this Lagenbucher device is according to the dissolution basic design. The screen is constructed such that medium flows equally through the entire cross section in a laminar flow pattern. Here also the flow is laminar flow pattern, not turbulent. And this is closed by secondary screen. Here two screens are there. F1 screen 1, F2 screen 2. And here one more is there in Langer-Bucher method. Filter is there. So by using this filter, we can prevent the undissolved drug from being eluted. So this is one of the provision in Langen-Bucher method. Next one, continuous flow apparatus by Tingstad and Regelmann. So this is consisting of glass cell that is of 6.1 centimeters long and 1.9 centimeters in diameter. Okay, and this is constructed with two small volume of glass filters. So these are the filters. Two small volumes of glass filter funnels are used. And this dissolution cell has a filter membrane that membrane will prevent the solid particles that is from reaching to the analyzer. And here one more provision is there for air trap. And here this is the analyzer. In continuous flow apparatus, there are external walls are there to prevent the control of excess flow of the solvent into the system. So here there is a provision for walls are there. These will control the excess flow of solvent into the system. The air trap. What is the use of air trap? The air trap is to prevent the air bubbles. Air trap is used to prevent the air bubbles. So the total system is maintained at a constant temperature, including flow meter, this lower filter, dissolution cell, and upper part of the filter, and air trap. So everything, the total system will maintain constant temperature bath. This complete assembly is immersed in a temperature bath that is maintained 37 degrees centigrade. So after that, we will collect the samples and we will go for analysis. This is description of continuous flow apparatus method. Next one, flow through modified column apparatus. This flow through modified column apparatus the figure here A, B, C, D, K, C, L, M, G. There are so many parts are there. So this is consisting of first one A. Where is A? Here. The A is solvent reservoir. This one is solvent reservoir. B is filter holder. This is filter holder. Next C. C here, here, here. These are all flexible tubings. These are tubes. Next, the D is pump. Next, E. Where is E? E here. This is filter holder. Next, F. Where is person? Here F is there. The F is support screen. This F is support screen. The lower one is this is E is filter holder. And G is there. What is G? This is polycarbonate cylinder. Here H is there. The H is nylon filter. F is support the screen. H is nylon filter. I is there. Here. I. The I is gasket. J is the parts of filter holder. This is also filter holder only. Next K. This K is standard tube. Next L. L is the flow cell. M is the receptacle. Okay. It's consisting of filter holder, and this is employing, the filter holder is employing 14-meter size of nylon filter, and this is attached to the flexible tubings. These are all tubings, no? So these will attach to the flexible tubing in the reservoir. So these are all attached to the reservoir. So here, keep the dissolution medium is free from particulate matter. Why? Because here different types of filters are present, no? That is the reason the dissolution chamber is free from particulate matter. So what is the use of pump? The pump is operated at variable speed. So this is capable of delivering the dissolution medium. The flow rate is 7 to 150 ml per minute. So this pump is allows to use of 0.1 normal HCl without causing pump deterioration. Okay. After that, the tubings are from the pump is connected directly to the bottom of the dissolution cell. Okay. So the filter in solvent reservoir keeps the screen is clean. The solvent reservoir in this filter is present. So that the filter will keep the reservoir clean. So after that, after analyzing, we will take the samples and we will go for analysis. This is flow through modified column apparatus. By using all these apparatus, we will collect the samples and we will go for analysis. Okay. These are all non-sink and sink methods. And next one is a continuous flow by Takanika method. The release of drug is immersed. So this is the assembly of last method, continuous flow apparatus by Takanika method. So here the labelings are there. First one is a intestinal simulated fluid reservoir. The B is the peristaltic pump, and C is the dissolution container. This one C, dissolution container. D is the water bath. E is the collecting reservoir. F is the motor. G is the pH electrodes. Where is G? Here. This is the dissolution medium. This is the pH electrodes. This is the basket. Next, F is the motor. This one is J. J is the pH meter. Okay. So the release of drug is measured with the aid of in vitro simulator device. This is consisting of flow type dissolution container. The dosage form is placed in the basket. So the dosage form is placed in this basket. Here provision is there. The basket is there. No, here we are mounting the dosage form in the basket, and we'll rotate the basket at the rate of 94 RPM. The dissolution medium volume is 300 ml. So the medium is removed by collecting the reservoir using peristaltic pump. So after that, aliquots are withdrawn using syringe and then filtered using Wortmann filter paper. And the same volume is replaced immediately with fresh medium to maintain sink conditions. So after that, we will go for analysis. So this is about continuous flow apparatus by Takanika method. This is the apparatus. Next, factors influencing dissolution and dissolution studies. One is physicochemical properties of the drug. Next one is the factors relating to dosage form. Physicochemical properties of the drug is drug solubility, particle size, polymorphism, solvates and hydrates, salt formation. Next one is the factors relating to dosage form. So here we need to study different types of excipients. All of you learn in detail about excipients part. All of you mentioned different types of excipient. like vehicles, binders, disintegrants, lubricants, colorings, surfactants, complexing agents, crystal growth inhibitors, flavoring agents. So, and also you need to write different examples for different types of excipients. So what we will conclude from dissolution studies? So by studying various factors that is influencing the rate of dissolution, and also we can optimize different properties of formulation. And also by conducting dissolution studies, we can know batch to batch reproducibility, batch to batch quality, and also we can predict in vivo performance of the formulations, and also we can estimate the solubility profiles of the drug. And also the best available tool is dissolution. By using this, we can quantitatively assure about the biological availability of the drug from its dosage forms, that is from formulation, that is in vitro dissolution condition. By using in vitro dissolution methods, we can predict in vivo performance of the formulations. Mean we can predict, we can have some idea how the drug is going to behave in the actual biological system with the help of dissolution. We can predict that. Also, we can predict the batch to batch variations and or lot to lot quality of the drugs. Okay, this is about dissolution. All of you follow reference books Brahmankar and Sunil Jaiswal. That book name is Biopharmaceutics and Pharmacokinetics. The publication is Valla publication. Already it is there in group, I think. Next one is the Leon Lackman. The dissolution methods are explained in detail in this book. And next one is Dressman Kramer. In this also the dissolution testings are there. And also Banker Mukma, Banker. In Banker, the explanation was very good. Better you read Banker Muk for this in vitro dissolution models.